I am Pranav Dhamane from Pragyaan batch 2020 of Bajaj Science Center, Wardha, here to present the topic Stages of Cell Division and Life Cycle of a Virus. Now, we will look after what is cell division. Cell division is the process by which parent cell divides into two or more daughter cells. Cell division usually occurs as a part of larger cell cycle. Now, here you can see how a parent cell is divided into two daughter cells. Now further, there are two types of cell division. One is meiosis and other is mitosis. Then, meiosis is divided into two stages, that is meiosis 1 and meiosis 2. First, mitosis. Mitosis is a type of cell division that results in two daughter cells each having the same number and kind of chromosomes as the parent nucleus, typical of ordinary tissue growth. It takes place in regular body cells and keeps cells living and growing. It also helps in healing the wounds. There are four stages of mitosis: prophase, metaphase, anaphase and telophase. Now before prophase, the cell prepares itself for cell division in phase known as interphase, and after karyokinesis, cytokinesis occurs. Interphase. It is the longest stage of the cell's life. It is the time spent between divisions. It produces all materials required for growth. It prepares the cell for division. Now, here we can see the first part of interphase known as G1 phase or growth phase of the cell cycle. The second part of interphase is known as the S phase, that is the synthesis phase of the cell cycle. In the synthesis phase, the DNA duplicates. The third part of the cell cycle is the G2 phase. It is just like a checkpoint that makes sure that the DNA is correct. The last part of the cell cycle is called mitosis and has four phases during which the cell divides into two cells. Prophase. It is divided into two parts, early prophase and late prophase. In early prophase of mitosis, the chromosomes get small, centrioles move to the poles of the nucleus and spindle fibers develop. Here, we can see in the diagram given below, pair of centrioles moving towards poles, chromosomes consisting of two sister chromatids, and formation of spindle fibers. Now late prophase happens when the nuclear envelope disintegrates and spindle fibers begin to move chromosomes towards the center of the cell. Metaphase. During metaphase, the chromosomes line up across the center of the cell, also called the equator or metaphase plate. The second phase of the mitosis is the metaphase. We can see the formation of metaphasic plate at the equator of the cell. Anaphase. It is the third phase of the mitosis, in which chromosomes are pushed and pulled towards opposite poles of the cell by the spindle fibers from the metaphase plate. In anaphase, the chromatids of chromosome move apart and travel to opposite ends of the cellular spindle. Here we can see the daughter chromosomes are separated from each other. Telophase. It is the fourth and last phase of the mitosis, in which the nuclear membrane and nucleolus reform. The cell prepares itself for final division. In telophase, envelope surrounds each set of the chromatid to form new nucleus and the cytoplasm starts to divide. In animal cell, cleavage furrow formation occurs, and in plant cell, cell plate formation takes place. Cytokinesis. In cytokinesis, the cytoplasm, organelles and nuclear membrane are evenly split and two new cells are formed. The two new cells exactly like each other are called as the daughter cells. Cytokinesis takes place when the cytoplasm divides and two cells with identical genetic material are formed. And now we will watch a short animated video on mitosis to explain each process in easy way. Now we will look towards the second type of cell division, meiosis. Why we are and who we are. We should find answers of these questions. Meiosis takes place in the gametes of an organism. People have a chromosome count of 46. When an egg joins a sperm, the count of chromosomes must remain 46. So the egg can only have 23 chromosomes and the sperm can only have 23 chromosomes, so that the integrity of the organism must be maintained. During meiosis, gamete cells undergo double division, maintaining the DNA count but reducing the chromosomal count to 23. When 23 chromosomes of a sperm and 23 chromosomes of egg combine, a fertilized cell having 46 chromosomes is formed. Now here we can see various phases of meiosis 1 and meiosis 2. At the end of meiosis, the individual gamete cell has divided from one cell to 4. Males produce 4 viable sperm and females produce 1 viable egg and 3 non-functioning polar bodies. Here we can see oogenesis, that is formation of a mature egg, and spermatogenesis, that is formation of mature sperm. Meiosis ensures that all living organisms will maintain both genetic diversity and genetic integrity. Meiosis ensures recombination of genetic material and formation of new varieties, and this leads to evolution. So now, here we have a short animated video to explain process of meiosis in a easy way. Life cycle of virus. Cycles involved in replication of viruses. There are two cycles in replication of bacteriophage. These are as follows: lytic cycle and lysogenic cycle. In the right side you can see the lytic cycle and in the left side you can see lysogenic cycle. 5 general steps involved in both lytic and lysogenic cycle are landing, attachment, tail contraction, penetration and unplugging, and DNA injection. Lytic cycle. In the lytic cycle, the viral DNA exists as a separate free-floating molecule within the bacterial cell and replicates separately from the host bacterial cell. Now here we can see the stepwise view of lytic cycle. First attachment, second penetration, third biosynthesis, fourth maturation, and fifth lysis. Now here is the cyclic view of lytic cycle. Now here we can see the lytic cycle graphically. Lysogenic cycle. Lysogeny is characterized by integration of the bacteriophage nucleic acid into the host bacterial genome or formation of a circular replicon in the bacterial cytoplasm. In this condition, the bacterium continues to live and reproduce normally. The genetic material of the bacteriophage is called as prophage. Sometimes the viral DNA gets detached from the host's chromosome and lytic cycle starts again. This is called induction. Induction involves either a spontaneous or environmentally induced excision of prophage from the bacterial chromosome. The phage that causes lysogeny is called temperate phage. This is also called host-guest relationship. Stepwise view of lysogenic cycle. First, viral attachment and penetration. Second, integration. Third, excision. Fourth, infection. And fifth, recombination. Now here we can see the cyclic view of lysogenic cycle. Now this image shows how both the cycles, lytic cycle and lysogenic cycle, are related with each other. Difference between lytic and lysogenic cycle. In lytic cycle, viral DNA interacts into host DNA. In lysogenic cycle, viral DNA does not integrate into host DNA. In lytic cycle, host DNA is hydrolyzed. In lysogenic cycle, host DNA is not hydrolyzed. In lytic cycle, productivity of viral DNA is high. In lysogenic cycle, productivity of viral DNA is low. In lytic cycle, virus is virulent. In lysogenic cycle, virus is not virulent. Lytic cycle occurs within a short period of time. Lysogenic cycle takes time. Lytic cycle does not have a prophase state. Lysogenic cycle has a prophage state. In lytic cycle, host cell is lysed. In lysogenic cycle, host cell is not lysed. Thank you for watching my presentation.